Good morning, everyone. Good morning. Hi, everyone. Good morning. Welcome to the class, to your own channel, 9th and 10th. I'm your teacher, Ankita. A warm welcome and a warm good morning to each one of you. How are you all? Yes, the class just started. Yes, Harini, the class just started. Good morning, everyone. Good morning. Yes, I'm perfectly audible and visible. That's amazing. Thank you for sharing this with me. Good morning, everyone. I hope that all of you had an amazing breakfast, not so heavy breakfast, right? I don't want you to feel sleepy during the class. I hope that you have your notebook with you and your pen, and of course, I hope that you have your water bottle with you. Yes. Yes, good morning, everyone. See, so many of you are here. I'm sorry if I miss out your names. Don't worry about it. Today, we'll be focusing on the 3 important chapters, or we can say the only 3 chapters we have in our syllabus for Term 2 examination. Yes, I hope that all of you are all set and ready. Very good. I can see that so many of you are here and all of you are in full energy and full focused. Good morning, everyone. Yes, those of you who have just joined now, I'm your teacher Ankita and we'll be discussing about the 3 chapters we have in bio for Term 2 examination. Yes, it's a marathon. Very good, everyone. Yes, so let's start our class without moving here and there, right? I hope that all of you have joined the Telegram group, right? This is a very important thing we have, right? Like you have been saying, ma'am, let's get connected on Telegram. And so here we are. So you can join the Telegram group link. The link is in the description box below. And over here also. Yes. And of course, don't forget to hit the like button for the video. It just means a lot when you hit the like button and we see the likes. And of course, ask your friends to join in the class, right? Tomorrow, on 10th of May, they also have their exam, so you can ask them to join. And subscribe to our channel. Very good. I can see many of you have joined. Yes. Okay. Very good. Now, there is one question which is there that, ma'am, how long this class would be? So, our target would be that, let's just have it for 1 hour or 1.5 hours, right? I'll not be holding you for a very long time. I don't want you to be here for a very long time. We'll quickly revise it. And then at 2 p.m. again, we'll be meeting for the quiz. Okay? Yes, I hope that I've answered your question that we will be meeting after some time, for the quiz also. We will try to finish up all the content that we have. Very good. Yes, I can see that many of you are new here. Welcome to the class and let's start, everyone. Good morning, everyone. Those of you joining right now, good morning, everyone. This is 9th and 10th channel, right? It's your own channel and we are discussing about the Bio Marathon. So all the 3 chapters, important concepts and some of the important questions. Very good, everyone. I'm so happy. I'm so glad. It's very grateful to each one of you that you all are here. I can see many of you feel that, oh, ma'am, we are scared. Oh, bio is really tough. No, we'll make it easy today. Very good. Ready, steady, and go. Let's start our class today, everyone. We have 3 important chapters, right? So if we talk about in bio, we have 3 important chapters. How do organisms reproduce, heredity, and our environment. So we are starting from the biggest chapter that we have, that is, how do organisms reproduce? Yes, everyone. Very good, very good. So again, I can see few of you are asking doubts. That's very good that you're asking doubts. And I would encourage you to ask doubt. But there's a small request that can you keep the doubts as of now, the chapter specific. For example, if you're talking about the chapter, how do organisms reproduce? So you can ask the doubts from that particular chapter. When we're moving to heredity, we can definitely ask the doubts from the heredity. And at the last, we have our environment. Yes? Awesome. Thank you for all your cooperation. Don't be tense, Kuldeep. We are here, right? I'm telling you, kids, if you are completely giving your 100% for 1.5 hours over here, right? You will not be missing any marks. Yes? Pay attention, learn along with us, all of us, and we are sorted. Very good, everyone. Very good. Let's start. I hope that all of you are ready. So let's start with the asexual reproduction. So we know that Reproduction is of two types. We have asexual reproduction. We know that asexual reproduction involves only one single parent. There is no variation and there is no gamete formation. So important thing about the asexual reproduction is this. One single parent, no gamete formation and no variation. The offspring will be very much similar to the parent, right? I hope that all of you are clear with this. Yes, we will be discussing about the difference between the regeneration and fragmentation. Very good. Okay, so let's start with the first asexual reproduction mode that we have is the fission. Now, of course, you can quickly write what you know about the fission, right? And I'll be explaining along with this. We know that fission is a type of mode of asexual reproduction where we'll see the splitting of the organism. So if the organism is splitting into two, we call it as a binary fission. The example of the binary fission we have as amoeba. The easiest example of it is amoeba and leishmania. Right over here, we can see the amoeba will first, of course, will undergo the nucleus division and then the cytoplasmic division. And then, of course, we'll have the identical daughter cells. This is really very important. The use of the word identical is important for all of us. Then, of course, we have multiple fission where, of course, we will see a formation of cyst. Inside the cyst, there will be multiple nuclei. And as soon as the cyst will burst, right, under favorable conditions, of course, these will grow into the new organism, right? Of course, we have the example in multiple fission as the plasmodium. Very good, everyone. So, I hope that this just sums up what is fission, right? Yes, I hope that everyone is clear with that. Splitting of the organism, fission, right? Of course, there are two words. Don't get confused. There is a fusion, that is mean combining together. Fission, right? Splitting. Very good, very good. I hope that all of you are clear with this. Very good, everyone. Yes. So, let's move ahead to the next type we have is a budding. Now, budding is the easiest one because why? We have the easiest example over here, that is the hydra. Now, in budding, what happens? We will see a small outgrowth of the bud, right? There's a growth of the bud. And the bud will get mature. And eventually, it will get separated out. And of course, we will see the formation of the new organism, right? Very easy. Very good. So that is the example of the budding, right? We have hydra. Of course, yeast is also is an example of budding. Very good, Bhagya. Yes. Very good. Very, very, very good. So we have these important things. Outgrowth is important. There's a formation of bud. Moving to the next, after budding, what we have? Yes. Very good. Everyone, a small request from my side. I would request all of you not to spam, right? You all are here, so you can focus on the learning part of it. You can pay attention to what I'm saying. If you have any doubts, then of course you can mention there. Yes? Very good. Yes, because if you will be typing a lot, you might lose the focus. Yes. Very good. Yes, that's amazing. Awesome. So budding is done, fission is done. Now let's move to the fragmentation. And I can see many of you are asking the difference between the fragmentation and the regeneration. So let's first understand what is the fragmentation. So in fragmentation, we know that there's an organism, right? A whole big organism. They will break into the small fragments. And these fragments will grow into a whole new individual. Yes? So over here we have the fragmentation. Example is a Spirogera, right? Will break into many fragments, and these fragments will be developing into the new organism. That is fragmentation. So are we clear with fragmentation? Give me a quick thumbs up. Diagram question can come up in your examination, right? Yes, there are very few diagrams we have in the bio part this time. So yes, it can come. Yes, Sukhjot. Very good. We will be discussing about the regeneration part once we are there. I hope that all of you are clear with the fragmentation. We will be discussing about that very soon. Very good. Now let's talk about the regeneration. And now many of you have a little bit of confusion around the regeneration. Yes, so what happens in regeneration is really very interesting. So in regeneration, if an organism is by an accident being cut into the small pieces, like an example of the planaria, each of the peas can grow into a new organism, right? Now over here, there's one important thing. Regeneration is not the primary mode of reproduction, right? So it's not that organisms will be using regeneration as the only mode of reproduction. This is a case if there is some accident or in very unfavorable conditions, right? Are we clear with this? Very good. So in the terms of the examples, you can mention about the planaria, right? You can mention about the regeneration happens in the multicellular organism or the bigger animals, where we'll see the regeneration of the tail and the starfish hand. Yes, everyone, are we clear with this? Yes, quickly give me a thumbs up. So I hope that the difference between fragmentation and regeneration is clear. Fragmentation, right? What will happen? Whole new organism is breaking into fragments and each fragment will grow into a new individual. Regeneration is not the primary mode of reproduction. If by an accident the organism is being cut, that part, right, over here in this case of planaria, that part will grow into a new organism. In multicellular organism or in the bigger animals, we will usually see not the whole growth of the organism, but a specific part. Like in the case of lizard, we will see the tail regeneration, and in the case of starfish, the arm regeneration. Very good. I hope that this is clear, everyone. Quick thumbs up. DNA copying will be discussing in the variation part of genetics. Yes. No, fragmentation is a mode of reproduction. Regeneration is not the primary mode of reproduction. Yes, I can see a lot of thumbs up. Everyone, keep the energy in the chat box, right? You can give me the thumbs up and these smileys. Very good. If you still have a doubt, please write in the chat box. We'll be able to take it up. Very good. I can see a lot of thumbs up. Amazing. Now let's move to the next part, that is the spore formation. This is a very important topic. Usually 2 marks question comes from the spore formation. So yeah, you can pay attention over here. Now spore formation is very interesting because what we'll see the spores formation. Now spores are there in the sporangium. This is important, everyone. Remember where the spores are in. They are inside the sporangium. Now sporangium is attached to a hypha, which is over here, right? And in the sporangium, what we have? We have the spores. As under the favorable conditions, what will happen? The sporangium will burst, right? And when these spores will land on a moist surface, they'll grow. Yes? This is nothing but the spore formation, right? A mode of asexual reproduction. And we see this in the bread mold or the rhizopus. Are we clear? Very good, very good. Yes. Spores are stored in the sporangia. Very good. Now I can see many of you are asking what is the difference between regeneration and spore formation. The first difference is that it's for these spores right over here. That's a primary mode of reproduction. Planaria, of course, is not the primary mode of reproduction. The way the reproduction is happening is definitely different. In spore formation, we have the spores that will be helping in the formation of the next generation. Whereas in the regeneration, the whole organism, if by an accident is being cut, will give rise to a new individual or the organism. Very good. Yes, Sujata, they are same, right? They are same. Hypha is a stalk-like structure onto which we have the sporangia. Very good. Yes, Divakar, I've answered your question. Very good, everyone. So are we clear with this? Very, very good. Yes, the very interesting question that in the fission will have the exact same size. They will be identical copies, right? In asexual reproduction, they will be producing the identical copies. Very good. I can see a lot of you giving me a thumbs up. Very good, everyone. Clear as diamond, clear as crystal. Awesome. Now that we are done with this, let's quickly take up a question. Now over here we have a question. Can you read the question? Quickly answer. Rajesh observed a patch of greenish black powdery mass on the slate piece of a bread. Now this question is from the sample paper. So we are doing the sample papers also along with that, right? Name the organism responsible for this and the specific mode of asexual reproduction. Quickly, everyone, this is so easy, right? We have just studied very good that the Rajesh saw on a bread we can see some black, greenish, brown, powdery stuff. That is nothing but the bread mold, Rhizopus, right? And that is nothing but the spore formation. Yes, it's a sample paper question. Yes, it is. Very good. So we have this answer, right? And if we talk about the marking scheme, right, it's very, very easy. You don't have to draw anything. You can just write in this way, and it's a question for 1 mark, and we have 1 mark in a pocket. Very good, everyone. So the greenish blackish powder that we have, of course, is the Rhizopus. Remember the spelling of Rhizopus and bread mold, right? Please do not write M-O-L-D. This will be wrong. M-O-U-L-D is a correct spelling, right? So remember this, everyone. Very good. So are we clear with this? Yes, Rhizopus is a fungi. We are talking about the spore formation. We're talking about the example that we have over here is the fungi. Mouse tail, I'm not sure. I haven't seen by myself. Yes? Very good. So I think still you have a doubt in the regeneration part. See, regeneration occurs in our body. Of course, we have the formation of new cells. Cells die, and of course, there's a regeneration of the cells. In us to happen, the regeneration process is really very difficult, right? We'll not be able to see by our eyes. Like if there's a cut on the arm, right? If there's a cut on the finger, after some time we'll see that the cells will come over there. That is a kind of a regeneration. We can see that, right? The cells are getting regenerated. But for that, in our body, to see the major changes, that's not possible. Very good. Very good. So are we clear with this? Very good, everyone. Here we have one more question, the second part of the question. Name its vegetative part and the reproductive part. We're talking about the fungi, right? So it's a very quickly easy thing that we have the hypha, which is the vegetative part, and the sporangia, which is the reproductive part. I think many of you are asking, ma'am, what is an hyphae? So over here we have. You can see over here, that is the hyphae, which is the vegetative part, and the reproductive part is the sporangia, which will have the spores. Very, very good. So we are clear with this part. A smiley for all of us. So the diagrams can come for you to draw also, and there can be a question based upon the diagram. So maybe they'll show you the diagram of a male or a female reproductive system. They might ask you to answer the particular functions of a particular labeled organ. Easy. Very good. Yes, Vriddhi, I have answered your question. Now let's move to the next asexual mode of reproduction. Yes, and that is the vegetative mode of reproduction. Now in vegetative mode of reproduction, of course, flower is not involved. The other parts are involved, like stem, root and leaves. Yes, everyone, are we clear with this? Very good. Very good, Krishna. Yes. Very good, very good. Hypha is a vegetative part, sporangia is a reproductive part. You're absolutely correct, right? Yeah, that's a very interesting question. If there's in some accident, if there's been cut, that's how they will be growing. Very good. So when we talk about the vegetative propagation, of course, 1 marks question can come and they can ask you to write the examples for it. So over here we have the examples. Propagation through roots occur in carrot and sweet potato. Stem occurs in potato and onion. And in leaves we will have the bryophyllum. Yes, very good, everyone. So the trick to remember this is that, see, we are talking about the propagation of roots. So I'm sure you must have seen carrot, right? It's deep down under the soil. So that's, of course, the carrot and the sweet potato. Now we're talking about the stem. We have the onion and we have the potato. Now if you look over here in potato, we'll see the buds and they will be growing into a new plant. And for the leaves is the easiest one, that is the bryophyllum. The advantage of the spore formation is that, of course, the growth of the organisms can occur on a very large scale. Yes, if they are provided with the right environmental condition, and in this case, that is a moist surface. Very good. So the advantage of vegetative propagation again is that, of course, it requires less of work, right? Of course, it can grow organically, naturally by itself. So if we have a potato, if you leave in a moist place, you will be able to see the buds, right? And if you take those buds out or the whole potato, put in the soil, we will see, we'll have a potato plant. Yes? Very good. Yeah, these are the few advantages of the vegetative propagation. It does not require much of the labor, right? Very good, everyone. So we are clear with this, right? Very, very good. Now, you asked about the tissue culture. Now, tissue culture is not there as such in our syllabus, right? It's there in the box. So, it's important for us to understand what is the meaning of tissue culture. In tissue culture, we'll be taking any part of the tissue, we'll be chopping it into the small pieces, adding it to the sterile medium, and of course, then transferring into the agar medium or the nutrient medium, and it will grow. Eventually, we'll have the plantlet, we will put it in the soil, and that's how the tissue culture will occur. Yes? So, this much information is well enough for all of us, right? Very good. Very good, everyone. I hope that you all are clear. Yes. So, now I can see a lot of you are asking about the questions which are not there as of now in our syllabus. Again, I would request you that your exam is just round the corner, right? So, let's focus on the syllabus that we have in our textbook. I understand that we want to learn more about certain things, but I would suggest that now it's not a right time. We can come back and we can learn together. But now, as of now, let's focus on the syllabus that we have in our textbook, that is the NCERT. Yes, everyone. Very good. So, are we clear, everyone? Quickly, a thumbs up. Very good. Yes. So, we can easily propagate the potato by putting in the soil. If I had the iBirds, it will be giving us a new plant. Very good. Okay, so you're asking me to again repeat the tissue culture. So, we will be taking the tissues, right? There will be a cut in the tissues. We will actually take a part of the plant. Tissue, specifically leaf, we can cut into small pieces. We'll transfer into a sterile medium. Then, of course, into the nutrient medium, right, which will actually help the plants to grow. There, of course, will be formation of the callus. We'll take it again in the nutrient medium. We'll add all the important nutrition. And from there, we'll be able to see the growth of the plants. That's a tissue culture for all of us. Very important. Yes, okay. Now that we are clear with this, let's move to the sexual reproduction. Now, are we clear with the asexual reproduction, right? Very good. Very good, everyone. Yes. Very good, everyone. And I can see there's very important thing that we have in the vegetative propagation, which is not so important in the examination, like in the CBSE. But let me just quickly take you through this grafting. Right, in grafting, we have two parts, stalk and cycon, which combines together, and of course, we'll be able to mix the characteristic features of two different plants altogether. We have cutting a simple, we can cut the rows and we can put it in the soil, and of course, a new plant will grow, right? Then, of course, we have, very good, yes. Then, of course, we have the layering process, right, which we usually do in the plant of a jasmine, right? Yes. Very good. So, all of these topics are really very easy. That's the information that we should remember about that. Very good. So, everyone, let's start and let's come back to the sexual reproduction. We have the flowering plants. Now, this is important, right? Now, I'm sure all of us are aware about the parts of plant. Yes? Very good. Lemon also, yes, very good. So, over here, everyone, I want your attention back on the main topic of sexual reproduction. Bhagyashree, we've just finished that. Give me a minute. I'll just come back on that particular topic again. But you can go back and you can watch it. Very good. So, over here we have the reproductive part, right? That is a flower in a plant. It has male reproductive part and the female reproductive part. Stamen is where, of course, we have the male reproductive part. Anther have the formation of the pollen grains, right? And over here we have the ovules in the ovary. Yes, we have pistil as a female reproductive part, which have stigma, style, ovary and the ovule. Very good. Yes. So, let me just take this interesting question. Yes. So, in grafting, we have two parts, stalk and scion, right? There are two different parts of a two different plant altogether. We will attach them together. That is the grafting. Cutting is simply, we have a plant, we'll cut it in a slant position, right, on a stem so that we can take it and plant it in the soil. That's the difference between grafting and the cutting. And what is stalk and scion? I have answered this question. Yes. Very good, everyone. Yes. So, are we clear with this part? Right. So, can you tell me where you will see the formation of pollen grains? Quickly in the chat box, where you will see the formation of pollen grains. Thank you so much, Shreya. Thank you so much. Yes, please write your questions. Yes. Everyone, quickly tell me where we will be able to see the formation of the pollen grains. Very good. That's the correct answer in the anther. And in the anther, we'll be able to see the formation of pollen grains. And over here only, we have the pollens. The pollens will move from here to the stigma. Of course, with the help of the pollination process, the transfer of pollen grains will occur, right? And of course, they will be moving inside the style, right? Let's quickly see a question and let's understand further. So we have mustard was growing in two fields A and B. We have while field A produced the brown colored seeds and field B produced yellow colored seed, right? If it was observed in field A, the offspring showed only parental traits of the next generation, whereas B have the majority of the offspring show the variation in the progeny. What is the probable reason for this? It's a very easy question, right? It was there in the sample paper. Yes, very good. So over here we have the case-based study, right? You've asked for it and we have this case-based study. So of course, if you look at this particular question, the answer that we will have is that of course in field A, the reason of the parental trait is because of the self-pollination, right? Because the pollen grains have been transferred from the same flower, right? From same flower to the same flower. That is the self-pollination. And in field B, of course, we have the cross-pollination. From one flower to the another flower of another plant altogether. Yes, this is the case-based study question. If you focus over here, this is a case-based study question, right? So we have two different parts, right? And this is how we can deal with it. Kuldeep, don't worry. Yes, CBSE is, NCRT is enough for all of us, right? For our examination, NCRT is the right book for all of us. Very good, everyone. I can see the self-pollination and the cross-pollination. Very good. Pistil, the female reproductive part, has stigma, style, ovary, and of course the ovules. In stamen, we have the anther and the filament. Very good. Now, let's talk about the fertilization process, right? We know that fertilization is nothing but the fusion of male and female gamete, right? Yes. So, we have the female gamete and of course the male gamete. They will fuse. Of course, we will call it as a fertilization. And there will be the formation of zygote. And when we talk about the another male gamete, it will fuse with the polar nuclei over here. And they will have the formation of endosperm. And endosperm provide the nutrition to the growing zygote. Are we clear? Yes. Yes, Rashmi. Yes, Rashmi, you're absolutely correct. Oldest goat, yes, we are doing the case-based study questions. So in polar nuclei, if you see, there are two nuclei, right? Polar nuclei. So we have two nuclei here that is in the center. So this, the male gamete, there are two male gametes. So one will fuse with the egg cell. That will form the zygote and one will fuse with the polar nuclei. It will have three nuclei, right? And of course, it will form the endosperm. Yes, yes, very good. Yes, revision notes will be enough for you if you have all the concept in your mind with you. Talking about this particular part again, all of you focus over here, right? Very good, Kalyani. 3N, yes, triploid, endosperm, very good. Double fertilization and triple fertilization, you guys are absolutely correct. So what happens over here? This is triple fusion. 2 times fusion is happening, right? Double fertilization first, fertilization is happening. Everyone, just focus here. First time fertilization is happening when the male gamete is fusing with the egg cell, and that will be formation, we will have the zygote. The second fertilization occurs when we have a male gamete fuses with the polar nuclei, right? It has 3 nuclei now. One for male, one from the male gamete, and 2 it already has. It will form the 3N, right? Of course, it will form the endosperm. Yes, very good. I hope that this is clear now. Very good. Open my books. Very good. Yes, 3C and 8N. 3, 7 cells and 8 nucleus. Absolutely correct. You're not late, Anu. We just started few minutes back. Yeah, 20 to 25 minutes back. Yes. Very good, everyone. I hope this is clear. Very, very good. So for the last time, I'm just repeating it again, all of you. Yes, so we are talking about the triple fusion that happens in the plants. 2 times fertilization is happening, right? 2 times fertilization. First fertilization, we have male gamete with the egg cell form the zygote. Second time, we have male gamete plus the polar nucleus form the endosperm. Very good. Yes, right? And of course, we have triple fusion because we have the 3 nuclei. 1 from the male gamete and 2 from the polar nuclei. Everyone, are we clear with this? Quickly give me a thumbs up. Quickly a thumbs up, everyone. Syngamy and triple fusion. You guys are already aware about it. Awesome. Yes, with the 2 time, when the 2 fertilization occurs in the flowers, right? We call it as a double fertilization. You can go in detail and mention about the syngamy and the triple fusion. Very good, everyone. Give me a thumbs up and we will move ahead. Awesome. Love to see the thumbs up. That means that all of us are confident and we are moving to the next question. Not question, but the next concept. So over here, after the fertilization process, we will see that there is a formation of a fruit, right? And a very common question asked, can you tell me what ovaries will develop into, right? And how will have the formation of seeds? Yes? We will be discussing about heredity. We will be discussing about heredity. Very good, everyone. Lots of thumbs up. Yes. Can you quickly tell me about that? The ovary will develop into what, and the seeds will develop, and the ovules will develop into what? Polar nuclei are a cell that is present inside the ovule, right over there, and they have 2 nuclei, 2 nucleus. Yes. Very good. Ovary will develop into the fruits and the ovules will develop into the seeds. Very good, everyone. You should remember about this. This is super important. Very good, very good. Claps for all of us. We're doing super amazing. Yes. Now that we are done with the flower sexual reproduction, we will be moving to the human reproduction, right? In human reproduction, we know that it's a sexual reproduction. Why? Because, of course, there's involvement of the 2 parents, right? Involvement of the 2 parents, gametes are produced and variation will occur, right? We know that variation will occur. Everyone, are we clear with this? We know that sexual reproduction is really important for the variation, right? Because of this only, sexual reproduction will see the variation in the next generation. Reproduction helps in having the continuity of the population of the various species of the animals, and of course, it actually assures that there is no extinction of the animals. Very good. Yes. Now that we are over here, let's quickly move and discuss about the female and the male reproductive structures. Yes, the reproductive system structures. Very good, very good. I can see crystal clear, crystal clear. Yes, I can see there is still doubt for the double fertilization. So you can go back and see. Just give me a minute. I'll just explain you very quickly in very brief. So double fertilization occurs in the flowers where fertilization is happening twice. Meet mandali. Yes. So what happens? First, the male gamete will fuse with the egg cell, having the formation of zygote. Then the male gamete will fuse with the polar nuclei and there is a formation of the endosperm. That is called as a triple fusion, and the first one is called as syngamy, double fertilization. Very good. Now let's talk about the male reproductive system. Now can you tell me the important parts of the male reproductive system, right? We know that sperms are produced in the testis. Yes, sperms are produced in the testis and testes are present outside the body of the male, right? They are present outside the body of the male. And why they are present outside the body? This is important for 2 marks, right? The scrotum is present outside because, of course, for the production of the sperm, the body requires 2 degree less temperature. Yes, so it's need 2 degree lower temperature than the body temperature. That's why in the scrotum, the testes are present. This is a very, very important question. Very good. I can see you're mentioning the names. Yes. So over here we have the importance of the scrotum and why it is outside the male reproductive body. Male reproductive, basically the body of the individual. Now if we talk about the different parts, we have the testis, right? Over here you can see the testis. We have the epididymis. We have vas deferens, which will carry the sperms from the testis to the glands that we have, seminal vesicle gland and the prostate gland. Now I'm just stopping here for a minute, right? I'm stopping here for a minute, Karthik. in the CBSE, we don't have the detailed parts of the seeds, right? So, I would request you not to focus over there as of now. Yes, Sukhjot, this is about the male reproductive system. Yes. Very good. So, everyone, can you tell me what are the function of seminal vesicles and prostate gland? Now, this is a very important question asked in the examination. I want you to answer it so that we all know that all of us are on a same page. Yes. Very, very good. Yes, we're talking about the parts, right? The function of the two important accessory glands we have. We have the seminal vesicle gland and the prostate gland. Yes, they provide the nutrition and they actually help in the transportation. Very good. Seminal vesicle provide the nutrition to the sperm, provide the lubrication. And of course, prostate glands. Very good. Yes. Very good. So, both of these glands plays a very important role in the providing the nutrition to the sperms and of course, help in the transportation. Very good. Yes, and NCERT is enough and we have the important function of these in our NCERT, and that's the, you can just mention about, it provides the fluidity, it provides a fluid, extra fluid for the lubrication. Apart from that, provide the nutrition and helps in the transportation. Very, very good. This is really important, everyone. Put a star over there in this particular topic. A very common question asked in the examination. Yes. Now, of course, once the sperm will be moving from these glands, they'll be getting the nutrition and the fluid, and of course, they'll be transported into the urethra. So, in males, there is only one passage for the urethra, for the urine and for the sperms, and they'll be moved into the urethra and through the penis, they'll be moving out of the male reproductive, male body basically. Yes, very good, everyone. I can see you have the clarity about it. Rashmi will be discussing that in the heredity part, but again, it's not so much in detail in your syllabus. So, we'll be just discussing the very basics for it. But you have to wait, we'll be discussing this in heredity. Yes. Variation is important for the survival of the species. Right, if the variation is not there, evolution will stop and eventually there is a chances of the extinction of the species. So, variation is important. Yes, everyone, I hope that this is clear. Quick thumbs up. Yes, quick thumbs up, everyone. Very good. Yes. So, we are talking about the male reproductive system over here. Important question are two. What is the function of the seminal vesicle gland and the prostate gland? We know that it provides the nutrition and of course help in the transportation. Next, we have why the scrotum is or why the testes are outside the body of the male. Because we know that for the production of sperm, right, there is a requirement of the 2° less temperature. That's why the testes are there in the scrotum. Very good. I can see a lot of thumbs up. With this, let's move to the female reproductive system. Right, we have the female reproductive system. Now, this is a very complex system as compared to the male reproductive system, right? Yes, very good. So, we know that the main gamete in the female reproductive system is the egg, or also called as the ovum. So, don't get confused, right? Egg or the ovum is produced in the ovary. Right over here, we can see in the ovary. So, ovary will be releasing the egg, right? And in the fallopian tube, it will be moving. Fertilization occurs in the, yes, fertilization also occurs in the fallopian tube. Can you tell me the another name of fallopian tube? Yes. Very good. I can see the answers. Quickly, everyone. All of you get involved. Very good, very good, very good. Yes, oviduct. Very good. Yes, Yashrat. Yes, that's the exact site. Yes, very good. Oviduct is another name of the fallopian tube. So, don't get confused in the examination, right? We know that fertilization will occur in the fallopian tube or the oviduct, and it is responsible for collecting the egg which has been released by the ovary. Very good, everyone. Then from there, the egg will move into the uterus, right? Now, uterus is the inverted pear-shaped structure. It is also called as a womb. If the fertilization occurs, of course, the zygote will get implanted over there, and of course, it will grow into the individual, right? And of course, once the time is done, like after in humans, 9 months, then it will be moving out from the mother's body. Yes, very good. Then we have the cervix over here, through which the sperms will be entering, and of course we have the vagina. Yes, And again, this is a very important part from where the sperms will be entering into the female reproductive system. Yes, very good Kartika, Avya, Krish. Kalyani, implantation will occur. Very good. Shaurya. Very good Metu, Gopal, Chitra. Yes, yes. The baby will grow in the uterus. If the egg is not fertilized, we know that the female body monthly will be going the process of the cycle called as the menstruation, right? The bleeding will occur, the endometrium walls of the uterus will shed themselves along with the egg, and that occurs once in a month. Very good. Yes. So are we clear with this, right? Implantation will occur in uterus. Very good. And the menstrual cycle will occur. Yes. Now let's talk about when the fetus is growing. There's a formation of placenta over here, right? As you can see, the placenta is there, and placenta plays a very important role in the transportation of all the important nutrient, right? And will carry the waste out from the fetus body, right? So over here we can see that's umbilical cord which is attached, and of course all the nutrient will be moving from mothers to the fetus, and of course all the waste will be moving back into the mother's body with the help of the placenta. Are we clear? Oxygen, carbon dioxide, all of it will be happening. Yes. Very good. So the main function of the cervix, of course, it basically allows the movement of the sperms through into the female reproductive system. Very good. So we are clear with it, right? Yes. So over here we have discussed about all of it. Now let's quickly take up a question over here. The question is, we have to trace the path of a male gamete, right, that takes place that fertilizes a female gamete after being released from the penis. So can we trace back the path? Yes. So we know that in the male reproductive system, what we have the formation of the sperms, right? And if we have to trace back in the female reproductive system, male gamete will travel into the female reproductive system after being released from the penis, will be entering into the vagina, then to the cervix, then to the uterus, and then towards the fallopian tube, right, where if the egg is, the fertilization will occur. Yes, everyone? Very good. Yes. Placenta is basically a part, right, we can see, which actually is attached to the mother's body, and of course it is also attached to the fetus with the help of the umbilical cord, and it actually helps in the transfer of the nutrient and the waste. Very good, everyone. Yes. So we have discussed about each and everything over here, and if this question comes, could be of this marks, right? And of course the B part of this question is, state the number of the sets of chromosome that is present in the zygote. Can you tell me how many chromosomes will be there in the zygote? The diagram is not compulsory in this question. Yes, diagram is not compulsory. So in most of the cases, they will ask you either if they want you to draw the diagram, they will ask you to draw the diagram. If not, then you don't have to worry about it. Very good, Ify. Coming back to the question, the number of chromosomes that we have in the zygote will be 23 pairs, right? Yes? Very, very good. 2 sets of the chromosomes or the 2n of the chromosomes. Either 23 pairs, right? Or 46 chromosomes. This is important, everyone. You have to mention 23 pairs of chromosome and 46 chromosome, or we have over here. It's a half marks question, so it's easy. Yes, very good, everyone. So with this, we are done with the how do organisms reproduce. The very important part we have in this is the yes, very good, everyone. Yes. So very important part we have in the how do organisms reproduce is of the contraception. Now contraception are those methods that actually will be helping in avoiding the pregnancy. Are we clear with this, right? They actually help in avoiding the pregnancy, unwanted pregnancy. Very good. So in one of that we have two important, of course we have various important methods. Barrier method is one where of course the easiest way, like the example of it is a condom, where the fusion or the meeting of the gametes can be stopped, right? So that's a barrier method. Then we have the hormonal method where we discuss about the pills, right? So these pills will be basically will be working upon the hormone level in the female reproductive system and will try to avoid the implantation or the fertilization process. Then we have the copper T IUDs, the copper T that can be placed inside the female reproductive system, that again will actually help to basically deactivate the sperms altogether. And then we have the surgical methods, right? And basically these are the kind of a permanent method where in the female reproductive system, fallopian tube will be cut and tied, and in the male reproductive system, the vas deferens will be cut and tied. Very good. Tubectomy and the vasectomy. Very good, everyone. So we are clear with this, and contraceptive methods are really important to avoid the unwanted pregnancies, and of course for the individual to take care of them from the STDs. Very good. Sexually transmitted diseases. Very good. So in males, vas deferens will be cut and tied, that is called as the vasectomy. Tubectomy occurs in the female where the fallopian tube will be cut and tied. Very good. So are we clear with this? Very, very good, everyone. So I can see that you are giving me a thumbs up. You are writing the answers. Awesome. Now with that, we are done with the first chapter, that is the how do organisms reproduce, and now we are moving to the heredity. So all of you who are talking or who are waiting for the heredity, so here we have. Yes, I'm giving you a break of 1 minute. Quickly drink water. Be here. Quickly drink water. I'll also drink, and we'll start with the next chapter, that is heredity. Female feticide, right? So in India and many other countries, what happens? If the parents and of course in the family, if they are not wanting a girl child, they will try to kill the girl child in the womb itself, right? That is called as a female feticide. Very good, everyone. Yes, all of you are ready for genetics already. I gave you a break of 1 minute. Estrogen, Lochin, is a type of a female reproductive system. Basically, it's a hormone, right? It actually plays a very important role in the sexual reproduction. Yes. Because of that only, we will see the various changes happening in the female body during the puberty. Yes, and progesterone is a very important hormone that plays an important role in during the pregnancy time. Very good. I can see we are talking about the father of the genetics. Yes. Okay, everyone. So let's start on that note. Let's move. Okay, let's move ahead. Yes. Let's discuss about the heredity. So I hope that all of you are clear. Yes, and now let's talk about the heredity. A very, very important chapter. A very important chapter. Oh, I can see you are asking about, so there will be some side effect of the hormonal pills. Yes, there will be. It's a bone. No, it's a muscle, right? It cannot be a bone. Yes. Very good. Okay, now let's discuss about the heredity. Now in heredity, what we have? Now, can you tell me what is heredity? Yes, Aditi, there will be the side effects. Very good. Yes, we'll be discussing about the laws of heredity. Yes, laws of inheritance. Why DNA is important. All of these questions will be catering now. Yes, Kuldeep, don't worry. We'll be discussing about it now. So can you tell me what is heredity? Yes, quickly, quickly. What is heredity? The answer is there in the word itself, right? It is nothing but the inheritance of the traits from one generation to the next generation. Like, of course, we have that, oh, your ears are same like as of your father or your mother. So that is nothing but the heredity or the inheritance that we pass the characteristic features from one generation to the next generation, and that we call it as the heredity. Very good, everyone, to the offspring. Now, of course, the characters like skin color, height, or eye color are usually inherited from the parents, right? We can get these characteristics from the parents. So this is heredity. Now if we talk about the heredity, and of course when we talk about the important part over there, we know that we have DNA, that is the deoxyribonucleic acid. Are we clear? We have DNA, and on DNA we have the genes, or we can say that the small fragment of the DNA is called as the gene, And the gene will be giving us a various different characteristic features like hair color, skin color, eye color, etc. So the genes are the one that actually carries the information or the traits or the characteristic features that we will be getting. Right? So DNA of course is present in all the cells inside the nucleus, and on the DNA, right, what we have the important characteristic features. A small part of a DNA called as gene that will be carrying the important characteristic features. So are we clear with this? Very good, Aditi. Blueprint of the protein formation. Yes, very good. Yes, it's deoxyribonucleic acid, the full form of DNA. Lavia, we'll not be able to define as it the female feticide. You can write it's the, you can mention what happens in that particular process. Yes, very good everyone. Very good. So we are clear with this, right? Very good. Now let's take up a question to understand more further. The question is, in asexual reproducing species, the trait X exists in 5%. See again everyone. Important question from the sample paper. Can we just focus over here? Very good. So in an asexual reproducing species, we have trait X that exists in 5% of the population, and trait Y exists in 70%. Which of the two traits is likely to arise earlier? Give the reason. So we have X and we have Y. There's a this trait is there in the 5% of the population, and this is there in the 70% of the population. We have to find out which of these actually arise earlier. Of course, we know that it's the 70% because it's there in the majority of the population. Whereas the 5% is there in a small population. Yes? So we can say that trait Y exists earlier as compared to the trait X, right? And of course, in the asexual reproduction, we'll have the identical copies of the DNA. So just make the same copy of it. Very good. Yes, everyone, Y, and it's a 1 marks question, and we got that 1 marks. Of course, if we write about the Y also, we can say that new trait that happens in the population is because of the sudden changes, right? Mutation is happening. Because of that, we have a lot of variation. So that's how we can write the answer. Yes, so now, till now we have discussed about the heredity, right? We have discussed about the genes. Now, there are important things. Okay, there is acquired character and there is inherited characters. So quickly tell me everyone, what swimming is or what good handwriting is. Is it an acquired character or is it a inherited character? Quickly everyone. So our Janvi genome will have all the sets of the gene that we have in the humans, right? Or in any other organism. That whole bundle of the genes, we call it as the genome. All the genes together. Very good. So swimming is an acquired trait that we'll be acquiring during a lifetime. It will not be transferred from one generation to the next generation. Whereas inherited trait like eye color, skin color, height, all of these are inherited traits that move from one generation to the next generation. A question can be asked upon this. Differentiate between the acquired traits and the inherited traits. So you know the answer now. Very good everyone. You're asking whether these disabilities transfer. So if it's a genetic disorder, it will be moving from one generation to the next generation, right? If there's a deficiency of vitamin C, parents don't have it, but it's kind that we have acquired because we are not eating a right amount of vitamin C, it will not be transferred from one generation to the next generation. Very good everyone. I can see the answers. And now, important thing. Can you tell me what we'll be learning now? Yes. About the laws, right? About the laws. Very good. I can see over there. No, even if you're late, welcome to the class, everyone. Those of you who are joining just now, I welcome you to the class. It's a bio marathon for the term 2 examination class 10. So join us. Don't worry, we've just started with the heredity. Very good. Laws, right? So over here, we'll be discussing about the laws. We're starting with the very first law that is easiest. That is the law of segregation. And we are talking about monohybrid cross. What is a monohybrid cross? Quickly. See, I'm asking you only the easy question so that you can quickly type. What is the monohybrid cross? Mono means one. Hybrid means a mix. A cross, we know that we are mixing a cross. Basically, there's a cross is happening. Yes? Monohybrid cross is a cross where only one characteristics we will be talking about. Mono means one, everyone. So in this particular cross, we will be discussing about only one characters, or one character will be considered. In a case of this particular plant, we are discussing about a tall plant and a short plant. Yes, over here, tall plant and a dwarf plant. After the cross, what we'll see that in the F1 generation, we will have a tall plant. Can I draw this over here? Yes? Yes, we have it over here. So we have this. Now, again, let me ask you, what is capital T and capital T? Very good, you're writing the cross over here. Super, awesome. So what is capital T and capital T? That's a dominating character, right? And small t is a recessive character. So the capital T will be dominating on the recessive character. This is really very important, right? It will be giving the plant the height, which is tall. And small t will give the characteristic to a height that is short, right? So capital T and capital T, small t and small t, capital T is dominating, and small t is recessive. Very good. Now capital T and capital T is called as homozygous. Can you tell me why it is homozygous? Because it has same allele, right? So these two are homozygous. What are these? These are homozygous. They are same, right? Capital T is here and capital T is here. That is homozygous. If we have capital T and small t, what we call this as? Yes. Very good, very good. If we have capital T and small t, what we call it as? Very good everyone. Pure breed is the capital T and capital T, or the small t and small t. Endosperm function, it actually provides the nutrition to the growing zygote. Heterozygous, very good everyone. Capital T and small t, if we have two different alleles, we call it as the heterozygous. So we are clear with this. Over here we have homozygous, pure breed, tall, and homozygous, dwarf. In the F1 generation, we'll get a heterozygous condition. What we will get? We will get a heterozygous condition. And if we do the selfing of the heterozygous, over here capital T and capital T, small t, capital T and small t, we will be able to see in the F2 generation, one will be pure tall, that is homozygous, two will be heterozygous, again tall, and one will be dwarf. From here, we can actually have two important things, genotype and phenotype. So, before we talk about this, what is genotype? Genotype will tell us about the genetic makeup of the organism. Yes? Are we clear? Genotype is what? It will tell us about the genetic makeup of the individual. Phenotype will tell us about the physical appearance of the organism. Genotype and phenotype. This is really easy. Very good. There's one question which is, I can see that we have allele, right? Allele is the alternative form of the gene. So over here we have a black hair color, right? We can have brown hair color also. This is the alternative form. Very good. So over here, the genotype of monohybrid cross and the phenotype of the monohybrid cross is over here. Phenotype is 3:1. Genotype is 1:2, 2:1. Are we clear? Yes. Now everyone just close your eyes and think about it. Monohybrid cross phenotype is 3:1. 3 are tall, 1 is short. The phenotype of it is what? 1 is pure tall, 2 are heterozygous, 1 is pure dwarf. Very good. Are we clear with this? This is monohybrid cross. Very good. Quick thumbs up everyone. A quick thumbs up. You can actually make the Punnett square in rough. You can. And if you want to, you can make in your main sheet also. Yes. Sukhjot, if the question asks you to draw the Punnett square, you can. Very good. I can see it. So that we have discussed about the monohybrid cross. Right? We have just discussed about the monohybrid cross, where we are discussing about one character. Over here we are discussing about the height. Clear as water. Wow. This just gives us so much of energy, right? Clear as water means that the concept is all clear. Amazing. Now let's try a question, everyone. Here we have a question. Again, if you look over here, Sahil is basically performing an experiment of the inheritance pattern of the genes. He crossed a tall plant, tall pea plant, and a short pea plant, obtain an all tall F1 generation. Interesting question. Yes. Look over here. Yes. Very good. I can see some of the trends that we have on various reels and other things. Yes. Everyone, focus on your examination. It's a very important time for you to focus on your examination and not anywhere else. Yes. So Sahil performed an experiment, right? And of course, we have the inheritance pattern, tall pea plant and the short pea plant. Yes. We have to find out what will be the set of genes present in the F1 generation. Yes, I can see the answers already. So we have tall, right, with a cross of small. What will happen? We will have the capital T and small t. Yes, that is the in F1 generation we'll have, right? So over here we have it. So that's 1 marks. Very good. The B part of the question is asking us, give reason why only tall plants are observed in the F1 generation, because capital T is dominating. Capital T, small t, it will be this, right? Because capital T is dominating, it will not allow the recessive gene to express itself. Yes, 1 marks in a pocket, yes. Very good, everyone. It is dominating because, of course, it's a characteristic features of it, and it will not be able to allow the recessive to express. That's a 1 marks question. Very good, everyone, right? And of course, if you want to draw it, you can write it in this particular way. This is the alternative way to write the answer. Very good. Okay. I hope that it's clear, right? And again, over here we have one more. And this is how we can mention about it. We can mention about the law of dominance. So the first law that we just discussed, let me just go back. Over here is the law of segregation. Yes, what is the law of segregation? Yes, recessive will get masked. Absolutely correct. It will not allow, the dominating gene will not allow the recessive to express itself. Yes. So we have the first law, there is a law of dominance that talks about the dominating allele, right, or the dominator characteristics will be suppressing the recessive characteristics. The second one is a law of segregation, right? We know that during the formation of gamete, the genes get separated from each other, right, and will carry only one allele of each gene. That is a law of segregation. We will be learning about the law of independent assortment in a while. Yes, so are we clear about the law of segregation? Remember the word segregation, separation, right? So the gametes will be getting separated out, of course, and one allele will be there for one particular gene. Very good. Segregation and separation. Very good, everyone. Awesome. Now let's move ahead and let's talk about the another. That, of course, we have it over here. That is another important law. But before that, we have a question, right? The continuation of that, we have that when the F1 plant are self-pollinated, right? The total of 800 plants were produced. How many would be tall, medium, or short? We have to give the genotype in the F2 generation. So how many of you know how to calculate this? Yes? See, we have discussed about the laws. 2 laws are done. Laws of dominance and law of segregation. Yes? These 2 laws are done. Can you tell me about the answer for this particular question? Yes? Very good. Yes, I can see that, right? Okay, Bhavya have mentioned the answers also. Very good. Let's quickly see how we will be able to, calculate it. So over here, as we know, there are out of 800, 600 plants are tall and 200 plants will be short. So we have 1:2:1, right, over here. And of course, we have this ratio. So when we talk about it, we can easily solve it in a way that we have 1:2:1, right? We have this particular part where we have capital plant, a tall plant and a heterozygous condition, right? So we'll add like 1 + 2 divided by 4 into 800. That will give us 600. And over here we have it 1/4 into 800. That will give us 200. Right? So that's the answer we have. Very good. 600 will be the medium And over here, this is the short. Yes. So allele are the alternative form of the genes, and genes are the one that will be carrying the information, right? So for example, information about the hair color, but the hair color could be different, right? It could be black, it could be brown. That will be the allele. Very good, everyone. Okay, so we have the last part of the question. We have to give the phenotype of F2 generation. So over here, if we talk about the F2 generation, it will be 1:1. This is the easiest part, because in F2 generation, like, we don't have much, right? And sorry, this is the F1 generation. And if we have it, this is how we can calculate. 400 and 400 is 1:1, so of course both will be 1:1. Yes, why are we dividing by 4? Because we have the 4 different types, right? 1:2:1. Very good. Yes, very good, everyone. So I hope that you all are clear with this, right? Yes, very good. So the easiest way to remember is that how many plants, how many species, how many new generation that we can see over here. Right over here we have the gametes, and how these are, right? 1, 2, 3 and 4. Very good, everyone. So I hope that this is clear. So if we talk about the calculation, let's just look over here. We have the heterozygous tall and we have the dwarf, right? Now the what will be the ratio of it is 1:1. Okay. Tall plant, of course, out of the 4 we have 2 which are tall, right? Of course we will multiply by 800, we'll get 400. And over here, short plants 2 by 4, 800. Of course we will get this 400. Easy. Yes, everyone. So I hope that this is clear. A quick thumbs up over here, everyone. A quick thumbs up so that all of us are there and all of us are there on the same page. A very important question. Yes, very good. Now let's move to the next part, of which is the dihybrid cross. Right? Now in dihybrid cross, a very important law that can actually help us is the law of independent assortment. Now the law of independent assortment states that the allele of 2 or more gene will get sorted out during the gamete formation, right? Gamete separation, and each of them will be expressing themselves independently. Yes? Yes. So we have over here that the more genes will be getting sorted out into the gametes independently of each other. This will receive one gene and does not influence another allele which is there. And we can actually see this particular law with the help of the cross, which is dihybrid cross. Yes. Yes. Are we clear, everyone? So over here we are talking about the law of independent assortment by dihybrid cross. Now can you tell me why this is called as dihybrid cross? Yes. Now dihybrid cross has 2 important traits, like we are talking about the 2 characteristic features, right? 2 characteristic features. Over here if we see, we have round seed and the other one is a wrinkled seed. The color of the seed is yellow or the green. Yes, 2 characteristic features. We will be going back to the question in a while. Just pay attention over here as of now. Yes. So we have the dihybrid cross, 2 different characteristics we are talking about. Yes. And these characteristics over here, if you see, this is a dominating one, and over here we have this is recessive. So in the F1 generation, what we'll have? We will have a round seed with the yellow color. Yes. Over here we have. Now if we do the cross, right, if the crossing will occur, the ratio of the phenotypic ratio of the dihybrid cross will be 9:3:3:1. Yes. Yes, everyone. So this is clear, right? So you don't have to worry about this cross, though it looks really very difficult. Yes, but it's super easy. Very good. Yes. So if you talk about it over here, we have capital R, small r, capital Y and small y. If we do the cross selfing of this, we will be able to see the ratio, which is 9:3:3:1. Very good. So this is how you have, and you can actually take a screenshot of this if you want to. Yes. Okay, let me explain you the law of inheritance, law of independent assortment once again. So this particular law states that the allele, right, or of the two or more genes will get sorted out, will get separated, right? But they are not dependent. It's not that if one allele is going over there, it will not, the other allele will not be able to express itself. Two alleles are not bound to stay together and express together. So the common example that I always use to explain is that, for example, you have your friend, right? So there are two friends. One of them wants to become an engineer and one of them wants to become as an architect, right? Do you think that the architect will lose its dream just because of its engineer friend? Yes or no? Let's take an example of you, right? Maybe you have an amazing friend, right? But both of you have a different career choices. It's not that where one will be going, you will be going along with it, right? You will be expressing yourself independently, right? So that is nothing but the law of independent assortment. Even though these genes or these alleles are there together, the expression of them will be independent. That will not be deciding whether, okay, if the one friend is going, the other friend will go. Yes? Very good. That is nothing but the law of independent assortment. Easiest way to remember this. Very good, everyone. Oh, I can see many of you are saying that we are NEET aspirant and of course will not be happening. Yes. Okay. Okay, very good. I think Karishma is sharing it. Yes, very good, Pratham. Yes, Aditi. Thank you so much. Yes, very good, everyone. Okay, now we have the definition of the law of dominance. You can quickly take a look over here and you can note down, right? We know what is the law of dominance. Now I can see there are few doubts over here, right? Let me just go back and let's just see that question first. 1 minute, right? We will go here and see the question. Not this one. Right. So I think you guys have a doubt over here. Yes. So all of you who had a doubt on this question, please pay attention over here. Okay. So how many plants we have? 800, right? Out of that, we are trying to find out that how many plants will be tall, medium or the dwarf, right? Yes. So we have this ratio, 1:2:1 ratio, right? We have one plant which is pure tall, two plant which is heterozygous tall, and one plant which is dwarf. So from this, how many plants we have? 1, 2, 3, 4. So are we clear why we have 4? Yes, everyone? Are we clear why we are using 4 over here? Very good. Then of course we have 1 plus 2. How many tall we have? We have 1 and we have 2, right? So total 3. 3 divided by 4 into 800 of course will give us 600, right? And when we talk about the short plant, how many short plant we have? Just 1, right? So over here if we 1 divided by 4 into 800, that will be 200. Yes? Are we clear? Very good. Very good, everyone. Yes. So I hope that this part is clear, right? Why we have 4? Because we are talking about 4 plants. Very good. Yes, I hope that this is clear and this is the other way you can write the answer. The other part is about the F2 generation, right? Over here we have. Why 1:1 over here? Of course, we have 1 and we'll be using the same thing. 4 different plants and that will be giving us the answer. Right? If the cross is happening between capital T and small t, we know that this will be this. Right over here. So what is the ratio if you look over here? It's 1 and 1, right? It's 50-50%. So that's why we have 1. Okay? And we have 2 by 4, 800, 400, 2 by 4 and this. Again the plants are 4, right? 1, 2, 3, 4. That's why we have 4 over here. Yes. So if you talk about this ratio, we have 1:1 basically. We have equal amount, right? We have 50% of the heterozygous tall and 50% of the homozygous recessive. Yes. Are we clear, everyone? Very good. Quickly give me a thumbs up and we'll move ahead. Yes. Very good, everyone. Yes, move ahead. Yes, we are there. Now, again, we have a very interesting question. Can you read the question? Though it's looked really very difficult. I can still see why we are writing 4, because we have 4 plants. That's why we are writing a 4. Yes, awesome. Now that we're done with this, let's quickly solve this question, everyone. We have two pea plants. One is round and yellow seed, and another is wrinkled and green, right? Produce the F1 progeny. We have this. In F1 generation, in F1 plant are self-pollinated. Which combination of characteristics is expected in the F2 generation? How many seeds will be there in the new combination of characteristics that will be produced when the total is 160 seeds, right, in the F2 generation? Yes. It came in your pre-board. So I'm sure, Mansi, you'll be able to answer this question. Yes, very good. So see, these are the important questions. Let's look at the solution, everyone. So over here, we are talking about the dihybrid cross. We have 30 round and 30 wrinkled seed over here. So first, of course, we'll find it out, and then, of course, we will calculate. Out of 160, we are planning, right? So we have 9:3:3:1 ratio, right? 3 we have over here. Number of round and number of wrinkled seeds is the 3/16, right? That is 30. Very good over here. And of course, the other part is that we have a new combination, and of course, it just shows the independent inheritance. Yes. Very good. So, you know what? For this, we can just count the number of seeds we have in the here. Yes? So, if you look over here, how many we have? That will be 3. Very good. Yes, if you talk about the pure thing, right? 9:3:3:1. The 3 that we have over here is helping us to find the answer. See over here, we are looking for round and green. Yes? Can we look over here? Round. This is green. Round and green. Round and green and round and green. How many we have? 3. Yes? Very good. And that's how we'll be able to find the answer. We will just simply multiply with the 160, right? And we will have the answer like this. Very good. And with this, we are done with this particular question, right? Now, moving to the important part of sex determination. Again, a very important concept from this question, and we'll quickly move to the our environment. Now, we know that in the males, we have X and Y chromosome, right? The sex chromosomes are X and Y, and in female, we have X and X. The other chromosomes are the autosomes. Yes. Yes, thank you, Mansi, for helping Alka. It's the F1 generation. Yes, absolutely correct. Yes, I am lefty. Yes. I'm sorry if I've forgotten you, but of course, you were there in our M class term 1 also. Yes. So, are we clear with this, everyone? So, we know that in the sex determination, we will be learning about a very important concept of the chromosome. So, we have X, Y and XX chromosome. These are called as sex chromosome, and the other chromosomes that we have apart from this, right, or the 22 chromosomes that are left are called as autosomes. Then, when we talk about that, who will be determining the gender of the kid, we know that it's the male who is responsible for deciding the gender, right? If the chromosome X, right, if we have the sperm with the X chromosome, and if it fuses with the female X chromosome, what we'll have? We will have X and X, and that will be a female. If the sperm carrying a Y chromosome fuses with the X chromosome, right? The progeny of the next generation will be XY, right? So we know that male are responsible for the determination of the gender of the next generation. Yes, very good. Yes, very good everyone. So we are clear with this, right? It's a sure short question. Usually asked in every year question paper. So please have this. Yes. Very good everyone. Even I am lefty too. Very good. I can see there are many lefties in the class. So everyone quickly give me a thumbs up, right? A very quick thumbs up everyone. I want you to quickly and we will move ahead. Now this chapter is done, right? And we will be moving to the next chapter, that is our environment. Yes. Very good everyone, that is our environment. You can easily study science in 4 days. Just follow our sessions, do a little bit of self-study, right? And you are through with it. Not a little bit amount of it, but whenever you're sitting, you just completely focus on the learning part of it. Very good. So we have the next chapter, that is our environment. Is the easiest chapter. 4 marks will be coming from this particular chapter. So here we go. What is an ecosystem? We know that an ecosystem have the combination or the interaction where the biotic and abiotic components like living and non-living components will interact. That of course will form the ecosystem. Biotic have all the living organisms, you, me, plants, etc. And over here we have the abiotic, land, mountains, etc. Okay, do you need a break of 5 minutes? No, break of 5 minutes will be too much, right? Yes, I can see there's a need of a break. Let's take a break of 2 minutes. Yes, this chapter is nothing but the laddu. Yes, it's a laddu only. So everyone take a break of 1 minute or 2 minutes and come back. Quickly come back, drink water. Yes. 2 minutes is it? Yes. Quickly everyone come back quickly. I'm just waiting for all of you here. Menti will be there at 2 p.m. Yes. So now we have studied. After this chapter will be over, we'll be done with all the chapters, right? And at 2 p.m. we'll be having the Menti quiz. Yes, the medium-sized plant are, we usually see them, right? But of course, when we talk in terms of the cross that we have over there, sometimes they're very heighted or they're very short, right? Enjoying the session. Thank you so much. I hope that all of you are, getting the right information for your examination. I'm also here. My break is done. Yes. Okay, everyone. Hello. If you're joining us now, welcome back. What is the importance of DNA copying? So DNA copying plays a very important role. If there is any mistake in copying of the DNA, there actually be chances of the various disorders, genetic disorders. So DNA copying is really important in the organisms. Yes. Awesome, everyone. So I hope that all of you are back, right? Call your friends again. Aajao. Let's join the session again, everyone. So we are discussing about the ecosystem. We have 2 types and 2 important components, biotic and abiotic components. Biotic are the living components and abiotic are the non-living components. The interaction of these 2, biotic and the abiotic, of course, happens in the ecosystem. Then, of course, one of the important thing in this is the food chain. Now, what is a food chain? The food chain, of course, is nothing but the sequence of transport of the matter and energy in the form of a food from one organism to the another. That is called as the food chain. Yes? So we have the plants, right? Plants are consumed by the herbivores, which are the primary consumers. Carnivores will be consuming the herbivores. Then, of course, one more will be consuming this. And that's how we have the various trophic level. Yes, and that is the food chain. Yes. The genetic disorder occurs if the genes, right, are being not, either they are missing or there is some defect in the genes. Yes. I can see you are asking about the 3 laws of the Mendel. We'll just discuss in the end. Or after the class, you can go back in this class. We have discussed all the laws. Yes. Okay. After the food chain, let's discuss about the food web. What is a food web? When there are few food chains which are interconnected, we call it as the food web. Are we clear, everyone? Yes, no issues, and you have no issues. You can go back and watch the session. Welcome to the class now. Yes. Why the DNA variation is important for the species? It is really important for the species to survive, right? For the survival, it's important, and of course for the continuation. So if there is a variation, right, the changes can occur and a particular species can adapt to these, changes that is happening in their surrounding. That's really important. So are we clear with the food web? Very good. I can see you're asking about the biomagnification, but before that, let's discuss about the energy law. The 10% energy law is 1 marks question, which is usually asked in the examination. We know that there's a 10% energy get transferred from one tropic level to another tropic level. 90% of the energy get lost in the various metabolic activities that happens in the body. So that is the 10% law of energy. So you can just note down this, right, that only 10% of the energy moves from one tropic level to the another tropic level. Yes. Very good. Yes, and that is the 10% law of energy. Over here we have a question. Let's see this question. So if we have in the food chain, we have these. We have green plants, we have deer and the tiger in the food chain, right? These arrows A, C and E indicates the energy lost, right? And these lines over here shows the energy transfer. So which of these three vertical arrows A, C and E, and which one of the two horizontal B and D will represent more energy transfer? Can we give the answer for this? Very good, everyone. So we know that A will represent more energy, right? Over here it shows like this. Energy is getting transferred as compared to A and C and E, and B over here will be more as compared to the D, right? Yes. Very good. Yes. So that's a question on the energy, law of energy, right? And of course you can write in this way. But I would suggest that when you're writing the answers, write in the points and not in the paragraph form, right? That's the one thing we have to remember about it. So over here we have this, right? Yes. Now let's talk about the biomagnification. You're talking about the DTT, my bad. So we know that biological magnification is a phenomenon where we know that the toxins will get accumulated as it moves from the one tropic level to the higher tropic level. Yes. So, for example, if a plant is there, right, and if there is addition of the DTT, which is a harmful chemical, right? If the water plants will be taking it, what will happen? Of course, they will be accumulating it. Then these will be consumed by the other animals. These animals will be consumed by other animals, and of course, the organism size is increasing, right? The organism size is increasing. In that way, what is happening? There is the accumulation of more toxin in the body of the organism. And that phenomena is called as biological magnification. There is an enlargement, right? The accumulation is happening more and more. Yes. DTT. Yes, so over here we are talking about the biological magnification. So it's a harmful chemical, right? I'm sure you must have heard about it. As it moves into any water bodies, let's just suppose over here in a case of this particular food chain, the plant will take it up, right? Of course, it will be going inside them. The toxin will get accumulated. If this plant was consumed by some other animal, the toxin will move into the other organism, but at a more larger scale, right? They will be getting more and more. That happens because of the increase in the mass of the organism. Yes? And then, of course, it will be more and more in the higher organisms. That is the biological magnification. Let's quickly see this example over here. DTT was sprayed in a lake to regulate the breeding of the mosquito. How it will be affecting the tropic level in the following food chain. So, we are talking about this, right? So, we have aquatic plants, aquatic environment, plankton, small plants, small fish, large fish and hawk. So of course if we add the DDT, what will happen everyone? We know that the amount of DDT will be increasing as we move from one tropic level to another tropic level. Yes. Artificial ecosystem में decomposers क्यों नहीं होते? We usually have it over there also, but amount is really very less. So in artificial ecosystem, we keep on cleaning, right? We keep on doing. We interfere over there. If you want to add, you can, but it will not be sustainable. It might stink a lot. Yes, very good. The definition of biological magnification, Devi. So over here we have biological magnification definition. Less energy, more magnification. What is the advantage of decomposers? Decomposers actually help in the breakdown of the dead and decay material, right? And of course, they'll be eating them up. They'll be breaking it down so that the environment can easily take them up. Yes. So over here we have the answer, and this is how you can write the answer. It's a non-biodegradable pesticide. It will be entered into the food chain. And as it moves from one tropic level to another tropic level, of course, it will be causing the harm. Yes, Ali, you are at the right place. You don't have to worry about it. You can just quickly go through this video, note down the important points, and I think you will be sorted. Yes, very good, everyone. Then, of course, in this chapter, we have a very important thing, that is the RR, 3 R's, right? Reduce the use of whatever we can, like reduce the use of plastics. We can easily use the glass bottles, metal bottles, etc. And then recycle. So this is important. I would say like 2 marks question can come from the waste management, right? Now waste management is really important because of course as the waste will increase, it will be difficult for us to manage it. So waste management plays a very important role in managing our waste, reducing the wastage altogether. So we have 3 R's: reduce, reuse and recycle. And then over here we have the last topic, that is the ozone layer. We know that ozone layer protects us from the harmful UV rays, right? And this ozone layer plays a very important role in helping our earth. Yes? Very good. Okay, so we know that ozone is O3, that is there over here, right? When the UV rays comes right over here, when there is a break of this, we have O plus O. And when this O will be combining with it, of course, we will see the formation of the O3. Okay, so this is important. Remember this, everyone. Now, if there is a damage to the ozone, of course, these are the sources that can cause a damage. We have CFCs, right? Chlorofluorocarbon. Very good. Yes. Chlorofluorocarbon, and of course, when we talk about the damage or the effects of the ozone, it can cause a skin cancer in the humans, eye cancer, right? It can damage the eye, skin, it can damage the crops, as well as it can actually harm the animals also. Yes. So the definition of the biomagnification is that it basically is a phenomena in which the toxin will get accumulated as it moves from one tropic level to the another tropic level. Yes. So mostly, Shashikala, usually we have either 4 or 5 tropic level because the amount of energy transfer will be really very less. Yes. We can regulate the biomagnification. It's very difficult to regulate it. The only way out is not to add the pollutant or add the toxin into it, into the water bodies. Yes. So over here we have a last question. Let's quickly look at over here. Gas A found in the upper layer of the atmosphere. Super easy question, everyone. We're talking about a gas A, right? We have the gas A, which is found in the upper atmosphere. It is deadly poisonous, but is really very essential for all the living organisms. There's a decline of it in 1930. We have to identify the gas. Gas is what? It's the ozone. We know that it's the ozone, or we can write it as the O3 also, right? The other part over here is asking that how we form. So oxygen is there. If the UV radiation will come, there's a break, right? We have 2 oxygen atom, and when this oxygen atom combined with oxygen molecule, we'll have the formation of O3. Yes. So are we clear, everyone? I'm not going into detail because it's a very easy and super important concept and you all are, aware about it. And the second part is that why it is essential for all the living organism, why this gas is really essential. And state the cause for the depletion of this gas. Of course, this is important. You can mention about that it provides the shield to the surface of the earth, right? It protects us from the harmful UV radiations. So this is how you can mention about it. That's the first part, and you can mention the second part in this particular way. That the chlorofluorocarbons or the CFCs will be causing the harm. Yes. And with this, everyone, we are done with our class, right? It's 12:30 now. We'll be meeting again at 2 p.m. Okay. We will be meeting on Menti, right? So over here we have, and the quiz will be there on complete syllabus. So you will be getting the Menti code there when you will join the class, right? Byju's have got you covered. Don't forget to hit the like button. Hit the like button, everyone. It means a lot, right? Share with your friends and subscribe to the channel. Ask your friends to join for the Menti quiz. And if you haven't already, join the Telegram group that we have. And we will be meeting. Okay, you have set an alarm. Very good. So why don't we clean the ponds? We do clean the ponds, but it requires much of the labor work, right? That's why. Cleaning an aquarium is really very easy. It's a 1 or 2 people task, whereas cleaning of a pond is a huge task, right? But it is done on a regular basis, right? But with a limited amount of resources. Yes, everyone. So I'll see you in class at 2 p.m. Right? Whatever we have studied, I know that bio easily. Just take a break of some time, right? Have some food. At 2 again we'll meet, right? And we'll have the Menti quiz. Take some time out. Quickly go through the NCERT chapters. Go through the pages, right? And come to the class well prepared. Yes. Okay, on that note, everyone, I'll see you in the class at 2 p.m. Bye-bye, everyone. See you in class. Bye-bye.